Hello everyone, my name is Ahmed Mustafa. I'm an assistant professor of pathology and lab medicine at the University of Saskatchewan in Canada. I'm also the clinical HLA lab director in St. Paul Hospital in Saskatoon, Canada. Today I'm going to give you an idea about cross-matching. So the main idea of this talk is to give you an idea about what we do and how we do it and why we do it. So this is an old paper published by Paul Terzaki and his team in 1969 in New England Journal of Medicine, and it's a retrograde study. And what they found that people who had negative cross-match were doing so well and their graft were functioning, as you can see from the numbers. 187 had a functioning graft compared to only 8 didn't have a functioning graft. On the other side, the majority of the patients who had a positive cross-match, they were also associated with graft rejection, as you can see, 24 compared to 6. After this publication, the field has completely changed and cross-match become mandated or mandatory before any kidney transplant has to occur. So the first early cross-match was done by a simple technique, and they called it the complement-dependent cytotoxicity, or the CDC, where they incubate cells from the recipient with serum from the donor, and they leave it for 30 minutes, and then they do multiple washes to remove the excess unbound antibodies, and then they incubate it for another 2 minutes with 5 microliter of a complement. And in the next couple slides we'll see what does the complement do. And then they leave it for 60 minutes, and then they add a vital stain, and they again look at the living versus the dead cells. So if the cross match is positive, then the cells will be dyed and will have a certain stain. If the cells are alive and they didn't die by adding the serum or the complement, then the patient will have a different dye. And then they count the living and the dead, and they decide if this is a positive cross-match or a negative cross-match. Few years later, they found the original technique doesn't work when you have a weak antibodies or weak binding antibodies. So they came with another idea where they can enhance this method by adding anti-globulin augmented. And they call this technique anti-globulin augmented complement-dependent cytotoxicity, or AHGCDC. In the next slide, I will show you the difference between the CDC and the enhanced CDC. But the idea here is they just added 1 microliter of the AHD together with the complement, and they leave it for another 60 minutes. So in this figure, it shows you how the CDC actually work. So basically, you can see in figure 1 here, the yellow is the HLA antigen present on the surface of the cell. And then when you add the serum from the patient, and if this serum has a specific HLA antibody that is going to bind to this antigen, it will bind. And then when you add the complement, if it found an antibody, the complement will bind. But if you look at figure 1, here the complement is not unable to bind, although the antibody bind to the antigen, but the complement cannot bind because the complement need multiple antibodies to bind first so that it can cross-link between them. So if you don't have a cross-linking, then the complement will not bind, and this will give you a false negative result. In figure 2, when you have couple or 2 antibodies or multiple antibodies bind, then now the complement can cross-link between these 2 antibodies and then can result in a positive cross-match. So in figure 3, this is what they did when to solve the problem in figure 1, where you add the anti-globulin antibody. This is what you can see in red. This is the enhancement, and this enhancement, it's an antibody is going to bind to an antibody. So if the antibody bind in the first place, this anti-globulin antibody or anti-human globulin antibody will bind to the same antibody, and then the complement will be able to cross-link between them. And in that case, this enhancement will solve the first or the issue seen in figure 1 in the presence of weak antibody, and will not give you again a false negative result. However, it's going to be positive because of the enhancement. So with every technique, there is some limitation. So let's see the difference between the false positive result and the false negative result in the CDC. So if we start with the false positive result, what's the causes? The first cause, if you start with a poor viable cell, like the cells has been already dying, and then you continue with this dying cells, and then you add your stain which detect the dying cell, this stain will stain them positive, so you will think that the cross match is positive. However, the positive was because the cells was already dying. The other thing is contamination of the positive control with the patient serum. So if for some reason someone just mixed up the positive control, which has to react positive with anything, with the patient serum, this can result in a false positive reaction. Or if you, instead of incubating them for 60 minutes, you increased the incubation, or you went for a lunch or a dinner, and then a couple of hours later you found that you exceeded the incubation time. In that case, this will also give you a false positive result because the cells will die from the excess of incubation. The other thing also, if it's an incorrect dilution of the serum, so if you usually have to do a pre-dilution or determine the dilution that you're going to use. If you use a high concentration of serum, this can result in a strong positive cross-match, which is going to be false. Or if you used a wrong temperature, like incubating them in an extreme cold temperature, the cells will also die from this extreme coldness. Or if the patient are taking some medication, some of the medication, like that there is some monoclonal antibodies, or some of the newer treatment that they use for like a biological treatment, all these treatment can affect the cross-match. On the other side here, if you look at the false negative result, it's usually the opposite of the false positive. So it could be the low incubation temperature, the short incubation time. If, for instance, you missed to add the anti-human globulin that I discussed earlier, or you forgot to put the complement, all this will give you a false negative result. Again, incorrect dilution, like if it's going to be a pretty diluted sample, this can give you a false negative result. If too many cells are added, then antigen excess can cause false negative reaction because antibodies now is going to be insufficient. Or if the complement that you started with is already defective and the complement is not binding, you have to do some quality control of the complement. Or again, if the patient are taking some medication as well. So what's the limitation of the CDC crossmatch that they made them look for a different technique? The first, this is less sensitive than other assays. As I mentioned before, they give you a lot of false negative or a false positive result. Also, if these antibodies that are not able to fix a complement, they will not be detected as a positive crossmatch because, as you know, not all antibodies are able to fix a complement. As you can see in the last example here, some IgG isotype like the IgG2 or the IgG4 or the IgA, they do not fix complement, and in that case it's going to give you a negative reaction. What if you have a non-HLA which is not harmful, like autoantibodies or non-HLA antibody? This will give you a false positive reaction because they can still fix a complement. So both HLA and non-HLA are detected. So if both of them are detected, you would not be able to know which is the HLA and which is the non-HLA because both will give you a false positive reaction. And for that reason, they decided to look for an alternative or a better technique to solve all these limitations. So the next technique that replaced the CDC is the flow cytometry, and most of the HLA lab now are performing flow cytometry for as a method for cross-match. Although there are still some smaller labs, they still do the CDC, but the majority of the lab are doing now flow cytometry. So what is flow cytometry? From its name, flow means there is a fluid in the motion, and this is why the cells that we injected into the instrument flow in a buffer until it become excited by the laser, and this is why it's called flow. Cyto means cell, and meter is mean measurement. So it's basically measurement the cells which run in a fluid. So what's the idea of a three-color flow cytometric cross-match? So in this figure, you will see that we incubate the patient serum, which could have some of the antibodies, with the donor lymphocytes. And here we usually isolate the lymphocyte from the rest of the white blood cells, because the lymphocyte has the highest percentage of HLA expressed on them, and this will increase the sensitivity of the cross match. So as you know from basic knowledge, T cells only express HLA class 1, but B cells express both HLA class 1 and HLA class 2. So the first step here is we incubate the patient serum with the donor lymphocyte. If the antibodies present in the patient serum find the prospective antigen on the donor cells, they will bind to them. The next step, we add a secondary antibody. This secondary antibody has a fluorescence dye attached to it. So if this secondary antibody bound to the primary antibody, we can see the fluorescence tagged antibody. And this will tell us that the primary antibody has bound, because if the primary antibody didn't bound initially, this secondary antibody will not bind and will be washed away. So the idea here is we want to see if the primary antibody has already been bound. And as you can see in this figure, the secondary antibody bound to the primary antibody, and because the secondary antibody has a dye, we can easily see this form of combination. The next step, we want to see which antibody bound to which cell. We want to know where is the B cells versus the T cells. And in that case, we add two other antibodies. One is CD19, which can bound to the B cells, and CD3, which can bound to the T cells. Both these antibodies are directly labeled. So in that case, if we can see the red dye, which is in the case of the CD19, we will know that this antibody bound to the B cells. If we see the other dye, which is the yellow dye, we will know that these cells will bind to the CD3 or the T cells. So if you see two dyes, green and red, you will know that this is a B cell positive cross match. If you see two dyes, yellow and green, you will know that this is a T cell positive cross match. If you see three dyes, you will know that this is both positive for B and T cells. And here is an example of a double positive, or both positive B and T cell positive cross match, because you can see that the CD3 bind, the CD19 bind, and the FITC binds. So when we look this by the laser on the flow cytometry, it will detect three dyes and will tell us that this is a positive B and T cell cross match. So this is the actual raw data that we see with the flow cell cross match. So on the left side, you can see the gating strategy. And because we used two different dyes that can isolate or can differentiate between the B and the T cells, we can actually, based on this dye, gate our cells or our population into B cells because it's stained by the CD19, or T cells because they are stained by the CD3, as you can see in the x-axis and the y-axis. Then the next step is we ask the computer software to look at the second dye present in the T cells, which is the FITC, or the anti-human globulin. And this is what you see in blue, the histogram you can see in blue. In the first square, or the first figure of the T cell cross-matching, you can see there is a red line. This red line represents our cutoff. So if the sample shifted, means that the samples is this T cells is already found another dye in it. This is why it's become shifted, and this dye comes from the antibody that bind to the primary antibody, or the secondary antibody FITC labeled. And you can see here, if you go in the next blue histogram, you can see there is a slight shift, and this is what we call a weak positive, or you can see in the third figure a strong shift, and this is what we call a strong positive cross-match. On the other side, we can also ask the software to look only on the B cells and then look at the second dye attached to the B cells. And it's follow the same exact scenario as the T cells. We put a red line, which is our cutoff, and if the sample has shifted to the right, this means it's positive. It's either could be a weak positive, as you can see, or it could be a strong positive. So in that example that you see, this is an example of both T cell positive and B cell positive cross match. So let's ask ourselves few questions just to make sure that you understand the concept of the flow cell cross match. So why we use T and B cell lymphocyte, and I mentioned this earlier, that both the T and the B cell lymphocyte has the massive expression of the HLA. So this will increase the sensitivity of the method. So which HLA is expressed on T cells? It's HLA class 1 only. Which LA is expressed on B cells? Both class 1 and class 2. So if you understand this concept, let's go to the next concept. So if you have a T cell positive, B cell positive cross match, what does this mean? Yes, both class 1 and class 2 are positive because they are expressed on both the T cells express class 1 and the B cells express class 2 and class 1. So if both of them are positive, this means both class 1 and class 2 are positive. In the next example here, you have a T cell negative and a B cell positive cross match. So what does this mean? It means that only class 2 are positive, because if class 1 are positive, T cell also should be positive. But since T cells is negative, then by exclusion it's only class 2 that are positive. So the third example here, a T cell positive and a B cell negative cross match. What does this mean? Okay, so this means more work is needed. And the reason why we need more work, because if a T cell is positive because of the class 1, B cell also should be positive because B cell also have class 1. So when we find something like this, we know that it could be a technical error, or we have to go and look for other causes like history of autoimmune disease, because autoimmune disease you have autoantibodies, and these autoantibodies could just bind to the T cells but don't bind to the B cells. Or history of monoclonal antibodies. Some of the monoclonal antibody treatment just target the T cells but doesn't target the B cells. Or history of vaccination, as we have seen few cases with patients who are taking vaccination, especially COVID, that this vaccine has altered the surface expression of some antigen and make the antibody bind non-specifically to this. So in most of the cases, that if it's just T cell positive, B cell negative, it could be technical or could be other reason, but it's not because of the it's not because the HLA is a positive T cell cross match. It cannot be only positive T cell cross match by. So why did crossmatch overcomes the CDC or the AHD CDC crossmatch? First, it's more sensitive because we use a lymphocyte which has a massive amount of HLA, so you can detect as small as number of antibodies. The other thing also, it can differentiate between the T cells and the B cells, and so you can know if it's a class 1 positive or a class 2 positive or both class 1 and class 2. And also it's the clinical correlation, as you can see, we will see in the next slide, that cross-match or the flow cell cross-match clinically correlated more with the clinical picture that we see with the patient. So in this one, it's a figure to show you the difference between the three methods of cross-match. The CDC cross-match, the CDC enhanced with the HG, or the flow cell cross-match. And what they have done here is they diluted the serum, and you can see in the x-axis the dilution factor. So if in the first figure you can see, or the figure which represent the CDC cross-match, you can see that when the serum was diluted 1 in 100, means you take 1 microliter of the serum and 99 microliter of a buffer, then the serum become diluted 1 in 100. You can see that the cross-match become positive. However, with the enhanced CDC, you can see with 1 and 400 dilution, the cross-match become positive, means the sample become more diluted, means the sensitivity increased more. So this means that if you take 399 of a buffer with 1 microliter of serum, you can still get a positive cross-match because the sensitivity increased. In the last example here, which is the flow cell cross-match, it can give you a positive cross-match with 1 in a 1600 dilution, means you take 1 microliter of the serum with 1599 or 1599 microliter of a buffer, it can give you a positive cross-match. So the more that you diluted the serum, the more, and then you detected with the method, this means that the method is more sensitive than the other method. And this is just to show you the difference between the three methods, and you can see that the cross-match or the flow cell cross-match is more sensitive than the other two methods. And this is a paper published by a group of scientists at the University of Manitoba in Canada. And what they looked at a retrospective study for 249 patients who has been transplanted between 1992 and early 2000. These patients were transplanted with a negative CDC enhanced cross-match, and they looked at their graft survival in these patients. Much higher rate of early graft loss rejection within 2 weeks and multiple rejection episode in the first 3 months in the group who was T-cell positive. These patients were transplanted, as I mentioned earlier, with a negative CDC, and this paper has proven that flow cell cross-match is more sensitive in detecting graft rejection than the CDC. So why we do cross-match? First, we want to avoid the hyperacute rejection, because in the presence of a preformed antibody that if we didn't detect it, this can result in a hyperacute rejection and the patient will lose the kidney. It also is important to detect the presence of antibodies in the patient's serum that can bind and destroy these potential donor cells, because this is a precious surgery. It can happen once or twice in a life, so you need to be very cautious in picking the right donor with which can match the right recipient. The other thing is the greater the degree of sensitization, the more difficult it is to find a cross-match negative donor. So if there is a huge number of antibodies present in the serum of the patient, then it's high likely this patient is going to react with most of the donors. So it's important to do a cross-match before transplant. The last thing, it's important to measure the risk of transplant, and to measure the risk of transplant is by doing a cross-match between the donor and the recipient. Again, each technique has its own limitation, and although flow cell cross-match is the current technique most of the lab use, we need to understand its limitation so that we can interpret it properly, the results. So first, it's not HLA specific, and as we will see in the next couple slides. Also, results of cross-match must be interpreted in conjugation with the antibodies of the patient and the typing of the donor, so that you can understand properly the result of your cross-match. Proper history taking, like patient history, the clinical condition, should be considered. You need to look at the whole big picture before reporting the result of the cross-match. So the main question here, do you think every positive cross-match is sufficient to decline a kidney offer? And the answer is not yes and not no. So we need to understand what's happening with the cells and the interpretation of the result. So if you look here at this figure here, this is a cell or a B cell, and you can see there are other receptors expressed on the B cells. It's not just HLA that the B cells express. Like, for example, it can express CD20, CD25, CD52 receptor, which is actually a specific targets for some of the monoclonal antibodies, as rituximab, that we will see in the next slide. Also, B cell receptor, the cells express B cell receptor. The cells also express other antigen and also express the FC receptor. So this other antigen could bind non-specifically to the IgM outside the cell, or it could bind to an autoantibodies, which is not harmful for the organs, but it can result in a positive cross-match. FC can result which bind to any immune complex present in the serum of the patient. It doesn't have to be HLA. So when you add your secondary antibody, this secondary antibody can bind to the B cell receptor and give you a false positive cross-match. It also can bind to the IgM and give you a false positive cross-match, or actually it can bind to the FC receptor and give you a false positive cross-match. So rituximab, what is rituximab? Rituximab is actually a drug that is commonly used in kidney transplant patient, and it's also used for other occasions like cancer cells and leukemia and other things as well. So let's understand what is rituximab. So rituximab is a chimeric monoclonal antibody. Chimeric means part of it comes from human, like the FC portion of the antibody, and the Fab come from the mouse, which is the other part of the antibody. And it's a monoclonal antibody. It can bind to the CD20 receptor present on the surface of the B cells. If you remember from the previous slide, you can see that the B cells express the CD20 on the surface. So because this is an antibody, it can bind to the CD20 present on the B cells and can result in a positive or a false positive B cell cross-match. So the function of this rituximab is to deplete the B cells from the circulation. So why we use it in kidney transplant? Because you want to immunosuppress the patient. You want to deplete the B cells from formation of antibodies, especially for highly sensitized patients. And also it's used for non-Hodgkin lymphoma, autoimmune disease, and or highly sensitized patient, as I mentioned. So how does the patient taking rituximab is going to affect the flow cell cross-match? Let's see it in more details. If you have a B cells and T cells from a donor, as you have seen in earlier in the slides, And then this patient is taking rituximab, so this patient has a lot of rituximab or monoclonal antibodies present in the serum. When you incubate the patient's serum with the donor cells, this rituximab present in the patient's serum is going to bind to the B cells, but it's not going to bind the T cells because T cells doesn't express CD20 cells or CD20 receptors. Then when you add your secondary antibody, which contain the dye, this secondary antibody will bind to the this primary antibody or to the rituximab and result in a positive cross match. This at the end will result in a decline of the organ. So it's very important to take a proper history from the patient before rushing into a result and saying that this patient has a false or a positive cross match. A proper history is very important to make sure that the report is going correctly, and in order to prevent an organ to be declined. So what is the type of cross-match or the types of cross-match that we do in the HLA lab? First, in order to know the type of cross-match, we need to identify if it's a living donor versus a diseased donor. And for living donor, we usually do initial cross-match, and this is the initial cross-match that we do during the assessment phase, where we get the HLA typing for the donor, the HLA typing for the recipient, and then we do the antibody screening for the recipient, and then we do actual physical cross-match to make sure that we didn't miss anything, and this is usually done in the initial assessment phase. Sometimes this recipient can stay for as much as 6 months or 8 months or 1 year or 2 years before they go to the theater for surgery, and for that reason we require another cross-match to be done, and we call this cross-match a final cross-match. This final cross-match is usually done between 2 to 3 weeks before surgery, and we want to make sure there is nothing new happening during this waiting period. In the other scenario here is the diseased donor cross-match, because this donor is more like a donor who died, and then their family member decided that they want to donate their organs for any recipient. So because we don't do initial cross-match, because this is usually happen all of a sudden, all what we do is a virtual cross-match first to see if this diseased donor is compatible with one or with any of our waiting lists, because these usually diseased donors don't come for a specific person. It comes and then we do a match. We look at all the waiting lists in our quota or our patients with the same blood group, and we start to look at each patient to see if this patient is compatible with this donor or not. We usually do this with a computer program. So this computer program has all the typing of our patient, and then when we add the typing of the donor, it starts to look at the antibodies present in the patient serum and see if this antibody is going to be matched to the diseased donor. And this is what we call diseased donor list, a diseased donor cross-match. From this virtual cross-match, you usually give you like a list of 20 or 50 patients that can match this diseased donor. Then we send this list to the physician on call, and then this physician on call or the transplant nephrologist on call usually pick between 3 to 5 of these patients and ask us to do a physical cross-match. And this physical cross-match, we call it a final cross-match. So it's very important to know the difference between the living and the diseased donor on the type of cross-match that we perform, and it's important to know the difference between the physical cross-match, which I discussed earlier, and the virtual cross-match, which I'm going to discuss very shortly in the next slide. So what's a virtual crossmatch? From its name, virtual means it's a software or a computer program, or it's not a physical crossmatch, means we don't actually crossmatch the cells with the serum. We just look at the antibody results and we look at the HLA typing of the donor and the recipient. So first, we look at the patient HLA antibody. So in order to qualify for the virtual crossmatch, we need to have at least 2 sets of HLA antibody done for the patient. And then we compare these antibodies to the donor HLA typing. So we want to make sure that the donor HLA typing or doesn't have an antigen that can bind to the antibody present in the patient serum. And we look at the presence or absence of DSA or donor-specific antibody, which is an HLA. So basically, what is the DSA or a donor-specific antibody? This is HLA present in the patient serum that has the ability to bind to the antigen present in the donor cells. And from that, we can predict the flow cross-match result or the physical flow cell cross-match result, because if it's a positive virtual cross-match, there is highly likely it's going to be positive with the physical cross-match. And the idea here of this virtual cross-match, like for example, if you have one donor, say like the example that I mentioned in the previous slide, a donor or a diseased donor, that the family decided to donate their organs to any patient on the waiting list. If you have 400 or 500 patients on the waiting list waiting for this diseased donor, it's impossible to do a physical cross-match with all these with all these patients on the waiting list. So the easier is just to use a software. This software has a program that it has all the antibodies of this patient, and once you put the HLA typing of the donor, it automatically link or cross or do a virtual cross match between this patient in the wait list with the donor HLA typing, and only give you the patient who are compatible. Like say from this 500 patient on the wait list, it can give you like maybe 50 or 70 who are possibly compatible with this donor or will benefit from this organ. In that case, you will send this 50 list to the physician, and then the physician will see which patient has the priority or has the best match of these 50, and then ask the lab back to do a physical cross match on maybe 3 or 5 of these patients. Again, every method and every technique in the lab has its own limitations. So it's very important to understand the limitation of each technique in order to interpret the result. So here, this is a paper published in 2009 where they did a correlation or a chi-square correlation between virtual cross-match and flow cell cross-match, which is the virtual or the physical or actual cross-match. So what they have seen here, as you can see in the blue square, 515 patients were both positive with the physical cross-match, were also positive with the virtual cross-match. But if you look on the right side, 28 patients were actually positive by the physical cross-match, but they were negative by the virtual cross-match. And the reason here why they were negative by the virtual cross-match, it could be just a false positive cross-match, as we mentioned earlier, because of the non-specific reaction, or could be a non-HLA antibodies, or patient taking drugs, or autoimmune disease, or autoantibodies that can result in a physical cross-match, but it's not it cannot result in a virtual cross-match, because virtual cross-match is only HLA antibodies that we added to the software. Another scenario here, they found that 83 patients who were negative by the physical cross-match were positive by the virtual cross-match. And the result here, it could be when the pathologist in the lab or the technologists in the lab, they overcall the antibodies. So they found like very weak reaction antibody, that this very weak reaction antibody or very low signal or very low strength of antibody, they call them. You don't just call any antibodies you found. You need to see if this antibodies are actual real antibodies or also has its own limitations. So if you overcall the antibody and then you add these antibodies to the software, then it's just going to give you a positive cross-match with every patient you add. So, and this is actually give you like maybe 13.8% false negative. It could be also due to allele-specific or denatured HLA epitope, which we are going to learn more details in the antibody lecture. Thank you for listening.